Hello friends, I am Varsha Gaitonde and here I am going to talk about gene drive, or that is also called as self-propagating genetic elements. Gene drive is a genetic engineering technology that propagates a particular set or group of genes throughout a population by altering the probability that a specific allele will be transmitted to offspring from the natural 50% probability. What will happen when we compare the gene drive or Mendelian population? As I can tell you, gene drive is against the Mendelian principle, or it is a deviation from Mendelian principle where the 50% probability of natural inheritance is altered. What happens in gene drive here? By altering or directing a particular segment forcefully to get into the segregated, or we can say in the future generations, even up to 99.5% of the odds alteration will be passed to the next generation. This edits the whole population without fitness or reproductive edge. When we talk about the Mendelian population or Darwinism, the basic principle is fitness, means survival of the fittest. When the genes or alleles are passed to the progeny, the fittest gene will survive and they propagate to the next generation. But genetic drive is against the fitness, means it favors selection coefficient rather than fitness. Genetic drive have been proposed to provide an effective means of genetically modifying specific populations and entire species. This technique may employ addition, deletion, disruption, or modifying genes. Genetic drive affect only sexually reproducing species, which excludes virus and bacteria, means that this technology cannot be utilized in virus and bacteria, only can be utilized where sexually reproducing species as well as higher eukaryotes, means it is highly successful in eukaryotic organisms. The basic principles how gene drive works means it is based on two things. One is it depends on the organisms which are having selfish genes, means we can observe or we can do this particular process in the organisms which are having selfish genes. That means ultimately the higher eukaryotes. Selfish genes means the genes which propagate themselves without any significance to that particular organism, means they propagate to a greater extent. There are some evolutionary significance, but it is not yet known. The second process is segregation distortion. Segregation distortion means the biasness in the passing of alleles to the next generation, or meiotic drive. Meiotic drive is also the same thing. Coming to the historical perspectives, first time gene drive was practiced or proposed by Austin Burt in 2003. Subsequently, but at that time that was not much popular because genetic engineering it was not that much successful. It was only boosted after the work of Esvelt and co-workers in 2014 who worked with CRISPR-Cas in the higher organisms. Even though CRISPR was proposed in 2012, it was limited to certain lower organisms, but in 2014 it was introduced to eukaryotes and higher organisms. The principle of gene drive, it works with sexually reproducing plants and animals with short reproductive cycles. It specifies target sequence to be cut and replacement sequence to be inserted. This system is specific to species, subspecies and alterations because as you know, the genome construction or the genetic constitution of different individuals is quite different. If we will talk about this at a species level or subspecies level, it differs entirely. Coming to the facts about gene drive, This can force a trait to a population, defying the usual rule of inheritance. That means, as I have already mentioned, it is against the Mendelian principle of inheritance. Gene drive guarantees that a specific gene will be inherited. In Mendelian population, it is unbiased, means the gene getting into the next generation, the probability is now 50%, but here it deviates from the 50% probability. Engineered genetic gene drive could alter the traits of entire population of organism. They represent a potential powerful tool for the sustainable management of ecosystem. How it works, it will be covered in the next slides. Gene drives are attached to the chosen gene and put into the organism. This particular mechanism is relevant to two particular fields of genetics. One is population genetics and the other one is allied sector of genetics, that is biotechnology. Coming to see how it works. Gene can have different versions. As you know, it is governed by different alleles. Each organism gets 2 copies of each gene, like chromosome. Without a gene drive, when parents with different versions of a gene pass on to their DNA, each version gets inherited by only half of the offspring. With a gene drive, when parents with different versions of a gene pass on their DNA, essentially all offspring will inherit the gene with the drive generation after generation. Coming to how it is aided by CRISPR technology. CRISPR system consists of 2 key molecules that introduce a change into the DNA. That is, as I can say, Cas9 and RNA guided endonucleases, Cas9 or CPF1. The another thing is RNA, that is guide RNA. 2 things are here. Enzyme called Cas9 or that is RNA guided endonuclease acts as a pair of molecular scissors that can cut the 2 strands of DNA at a specific location in the genome so that bits of DNA can then be added or removed. In the second case, a piece of RNA called, or in the second step, here a piece of RNA called guide RNA consists of a small piece of pre-designed RNA sequence of about 20 bases long, which is located within a longer RNA scaffold. The scaffold part binds to DNA and the pre-designed sequence guides Cas9 to the right part of the genome. This makes sure that Cas9 enzyme cuts at a right point in the genome. This means how the mutation process occurs. Whenever the mutagen mutates or it affects a particular, means when deletion happens, that further process will be how the recombination occurs, or we can say that particular repair process will occur and that particular sequence will be recovered. That is how the guide RNA works here. And after guide RNA process, with the help of different enzymes, that will be recovered. The guide RNA is designed to find and bind to a specific sequence in DNA. Guide RNA has RNA bases that are complementary to those of the target DNA sequence in the genome. This means that, at least in theory, the guide RNA will only bind to the target sequence and no other regions of the genome. Cas9 follows the guide RNA to the same location in the DNA sequence and makes a cut across both strands of the DNA. At this stage, cell recognizes that DNA is damaged and tries to repair it. Scientists can use the DNA repair machinery to introduce changes to one or more genes in the genome of a cell of interest. This is how the guide RNA and the RNA guided endonuclease works with the principle of homologous recombination by cut and repair mechanism. This is how the mechanism goes. In Mendelian population or normal inheritance, what we'll see, every generation passing of that particular gene is 50%. Here you can see in the left figure, one copy is inherited from one parent, and that is 50% chance of passing into it. In the second figure you see where the gene drive operates. Here gene drive is crossed to wild type, and by that, when one copy of the gene is with gene drive and its homolog is with wild type, by homologous recombination, that is the principle operates in CRISPR-Cas, where the cut and repair mechanism of natural repair process in any DNA or any genetic material, it gets the same copy of 2, and that is how it makes 100% chances of passing into the progeny. That means particular kind of the combination will be passed and propagated in the next generations. That is how it is showed here in random inheritance. The number of that particular type of gene will be limited, but in case of biased inheritance or gene drive, the chances where we introduce a change that will be propagated in the further generations. How it spreads in a population. As the genotypic frequency can never be more than double in frequency with each generation, it needs dozens of generations to affect a substantial function of the population. That means when we are altering a single individual, means to propagate and spread into the population, it takes many generations. But when we introduce the same mechanism in different organisms or several organisms, maybe thousands, means it may take hardly 10 to 12 generations in a moderate populations to spread across. The fixing of gene drive in the population, it depends on 3 major factors. One is individual fitness, then rate of allele conversion, and population structure. As you might have studied in the population genetics, how the genotypic frequency changes generation after generation, and based on the fitness, allele conversion and population structure are the basic things how the gene drive work in the population. In a well mixed population and with realistic allele conversion frequency, maybe 90%, population genetics predicts that gene drive gets fixed for selection coefficient smaller than 0.3. That means the reproductive ability should be limited to 30%. In Mendelian genetics or normal Hardy-Weinberg population, we expect the maximum reproductive fitness, or we can say the maximum reproductive capacity as fitness. But here the thing goes in opposite direction. As I already told you, it works with the selection coefficient, where the reproductive success should be limited to 30% or nearby. This is in contrast with normal genes, which can only spread across large populations if they increase fitness. Coming to the applications of gene drive. Gene drive pave a way towards sustainable agriculture by reversing pesticide and herbicide resistance. Example, glyphosate resistance in weeds. As I have already told you, it is against the fitness of an organism. That means whenever a weed wants to overcome or create resistance towards that particular glyphosate, which is a post-emergent herbicide, that can be controlled. Gene drive eliminates diseases like malaria, dengue, yellow fever, Zika, then sleeping sickness and other by altering insect species to no longer spread them. This is also the same principle like selection coefficient will operate here, given more importance. Then eradicate invasive species. Invasive species means the species which are of not important, and it may be a predator which are causing harm to the organism which are helpful to the human being. Gene drive works against fitness and favors selection coefficient or keeps the population size under control. Coming to the challenges for gene drive, it must be able to cut the target gene. This is quite difficult to generate or to sequence or to make a target particular Cas9 protein, as well as the RNA guided endonuclease for each species or subspecies to target a particular gene. The second challenge is it should not harm the organism's ability to reproduce. It has to be accurately copied. It has to be evolutionarily stable. Copying the drive isn't the only way the cell can repair the break. It can just jam the broken DNA ends together. Limitations of gene drive: it requires many generations to spread through population. It can only reliably edit genes and sequence that are important for the organism's survival and reproduction. Sequences that aren't important can only be removed, not edited. It can't alter a sexually propagating population and will have trouble with species that can reproduce with or without sex, like many plants. Some type of alterations would need to be continuously reintroduced. Example, driven trait that is somewhat harmful to the organisms will eventually break. Thank you. Here I would like to mention one more advantage of gene drive. When we lose some species either in the extinction or genetic wipeout, there also we have a chance to get it back, as this goes against the evolution or evolutionary significance. I hope you liked this video. If you have any doubt, you can put it into the comment box, or if you have any suggestion, you are always welcome. Thank you.